students. Today we will study the cell structure in algae. On the basis of their organization, algal cell may be differentiated into prokaryotic and eukaryotic or mesokaryotic type. The prokaryotic cell organization is found in Cyanophyceae, which is characterized by the presence of incipient nucleus, absence of membrane bound organelles like plastids, endoplasmic reticulum, Golgi bodies, and mitochondria. The absence of basic protein like histone in DNA and the presence of mucopeptide in the cell wall and the absence of mitosis. A eukaryotic cell, on the other hand, is characterized by the presence of well organized nucleus and membrane bound organelles like plastids, mitochondria, and Golgi bodies. Majority of algae shows this type of cell organization. An intermediate type of cell organization like mesokaryotic is found in the member of Dinophyceae, where although the nucleus has a nuclear membrane and the chromosome eukaryotic character, basic proteins are absent, as in case of prokaryotic character. The cell wall. Cell wall, most of the non-motile unicellular and multicellular algae possess a typical cellulosic cell wall of non-living material outside the cytoplasmic membrane. It is composed of pure or mixed carbohydrate, cellulose, mucilage, and pectide, etc. And the substance like alginic acid, fucoidine, fucin, and hemicellulose are present in the brown algae, that is in Phaeophyceae. Pectin is pectin in red algae, Rhodophyceae, calcium carbonate in coralline algae, and silica in diatom in Basilarophyceae. Electron microscopic studies have revealed that cellulosic cell wall is composed of microfibrils of cellulose which vary in thickness from 30 to 200 micrometer. The microfibrils remain embedded in a smooth or a granular matrix and are variously oriented. They may be arranged in two layers at right angle to each other in a three layer or in a random manner. The cell wall of diatom is more complex, having pore with a perforated membrane. In the blue-green algae Cyanophyceae, the cell wall is mucopolymeric, mainly consists of glucosamine, amino acid, muramic acid, and diaminopinamalic acid. The true cell wall is, however, absent in some algae like Euglena, Gymnodium, etc. They simply have a bounding material of cytoplasm known as pellicle. Flagella. Motile vegetative or reproductive cells are found in all the group of algae except Cyanophyceae and Rhodophyceae. These motility is due to small filiform thread-like protoplasmic appendages called flagella. The number of flagella varies from 1 to 4 to many in Woodogonium and Vaucheria. They are mainly of the following two types: whipless or acronymatic flagella. Such flagella have a smooth surface. Tinsel or plurinymatic flagella. The surface of these flagella is covered with fine filamentous appendages known as mastigonemes or phlegma. They are further divided into three categories on the basis of the arrangement of mastigonemes. Pentonymatic. In this type, mastigonemes are arranged in two opposite rows or shows radial arrangement. B. Pectochrononematic. In pectochrononematic flagella with a terminal fibril is known as pectochromonematic. C. In stichonomatic, here mastigonematic develop only on one side of the flagellum. A motile cell may have either one or both the above mentioned type of flagella. It is a specific character. If all the flagella of a cell are similar, it is known as isonot. When dissimilar, it is known as heterocon, isocon.